Welcome, everyone. Welcome to the first in line, the new TN2 webinar series for this academic year, which is actually centered around the Amsterdam Neuroscience Research Programs. My name is Misha Wilhelmus. I'm a neurobiologist and clinical pharmacologist at the Department of Anatomy and Neurosciences here at the Amsterdam University Medical Centers. I'm your host for today. And my co-host is Erik Reitz. He's a professor in cellular imaging at the Department of Molecular Biology of Amsterdam University. And he's a program leader of the research program, Cellular and Molecular Mechanisms. So welcome, everyone. This is a Zoom session, so it's a webinar by Zoom, meaning that the speakers and the host are visible to you. You cannot be visible yourself, and you are also unmuted. There are a couple of options in the slide bar. So you have a chat function. The chat function is actually only meant for the organization, so we can chat with each other if something goes wrong. But if you, of course, want to participate and have a question, or you want to upvote questions of other attendees, you, of course, can do so with the Q&A button in the slide. So hopefully you will see them. And at the end of every presentation, so we have two presentations today, we will, together with my co-host, select popular and, of course, also interesting questions, and those will be addressed by the speakers. So in this Zoom session, you also have some control yourself, of course, and that is the way you actually visualize this presentation. So you have the shared slides on the one hand side and the presenter on the other, and the way you want to adjust both, you can do yourself, and you can do this by clicking on the boundary between the shared slide and the actual speaker screen, and you can adjust right or left, how big or small you want to have one or the other. So you can adjust the size accordingly. So it's a Zoom side-by-side view. If you want to adjust, please do so. So TN2, what is TN2? It's the Translational Neuroscience Network, and it aims to translate between fundamental and the clinical science. And every year we have an annual conference, and this last year we started with webinars during the Corona crisis, and it was a very great success. So we'll keep on doing that for this year again. So these webinars are focused around the theme of the TN2, so neurology, the field of neurology, but also psychiatry. And the idea is that, of course, academia intermingles, but also an opportunity for academia to meet industrial partners and explore collaborations. So I already mentioned last year we had also a webinar series of TN2, but this year we are centered around the 9 research programs we have in Amsterdam Neuroscience. So of course, 9 programs meaning 9 webinars, and every month, starting with this today, until June 22, we have a new, during lunch webinar. And the idea is every time similar, we have a cross-organizational setup, meaning one presentation of some key speaker, a key speaker from the Amsterdam Neuroscience Network, and then presentation of one external excellent speaker. And again, today we have such an excellent speaker. So it's free to join, and please spread the word if you know anyone or any organization who would be interested in joining our webinars. So without further ado, I will give the word to my co-host Erik Reitz, who will elaborate a bit more on the research programs of the neuroscience.
Thank you, Misha. Also from my side, a warm welcome to everybody who's joining now this lunch session from the TN2 meeting. Also warm welcome to Aniko and Joshua for Sophie for the presentations later on. A short background on the research program, because it is a relatively new program, the cellular and molecular mechanism, but also a real essential one, because as you can see in the vertical columns, many of us and you are working on different topics, neurodevelopment, neurodegeneration, inflammation, neurovascular, But what is at the heart of many of these disorders and also in the development are the cellular and the molecular mechanisms. And this is what we share, what also a lot of the PhD students, diploma students, postdocs also share in their type of experiments. And this is something that we have in common. So that is why we deliberately have made this group on the cellular and molecular mechanisms, ranging from the genetics to molecules and proteins, but also the life science enabling technologies that are involved in here, from omics to microscopy. So this will, I hope, really form the heart for a lot of scientists in Amsterdam neuroscience and others that we can share the expertise and learn from each others. So that's on my half. Misha, should I give the word back to you, or give the word immediately to Aniko so that you can start with the hardcore science part?
Well, let me first show everyone the program and the titles of the presentation. So Aniko Garozzi will start with early life stress-induced cognitive decline and a role for the fatty acids and neuroinflammation. And after that, at 12:50, Sophie Laye will start on give presentation on dietary polyunsaturated fatty acid and the neuroimmune interactions of and the role of oxylipids. But before doing so, let me just briefly introduce both speakers. So Dr. Aniko Garozzi is Associate Professor at Swammerdam Institute of Life Sciences at the Center of Neuroscience. And she leads the Brain Plasticity Group at the University of Amsterdam. So she was a postdoc at the UCI at the lab of Dr. Barham, where she studied how enriched early life experience rewires the hypothalamus. And at the end of 2010, she started her own team, and she's currently an associate professor at the University of Amsterdam. Her research is funded by several national and international programs, and her group includes both postdocs and PhD students, of course. Her research focuses on programming of cognitive functions by early life stress and aging, and on the role of the metabolic signals like nutrients and epigenetic mechanisms in this context. Her work encompasses both preclinical work on established mouse models of chronic early life stress, but also clinical work, and she's interested in the development of peripheral, as in neutral, interventions to prevent and even maybe reverse the lasting consequences of early life stress, as well as human studies investigating the effects of stress on breast milk composition. So that is the first speaker. She will start briefly. And then I will also introduce again the second speaker, just to keep you here and stay for the international speaker, because that is very important. She's actually the research director at INRAE. She's the head of the Nutri-Neuro Institute in the University of Bordeaux, but also she's the head of the OptiNutriBrain, which is a collaboration between Bordeaux University and Laval in Canada, and the international research network Food for Brain Health, which includes 13 partners of both France and Canada. Her scientific career is devoted to understanding how nutrition contributes to mood and cognitive disorders, focusing on the effect of food and nutrients on brain functioning using integrative and translational approaches. Her research aims at defining a healthy nutrition for the brain, finding reliable predictive biomarkers of cognitive and mood disorders, and new food-derived bioactive molecules in the brain to develop innovative nutritional and pharmacological strategies to prevent and treat brain diseases. And she's already been awarded with several academic achievements, which the one most prestigious is, and I hope I pronounce it correctly, Lauriers de Laye in 2015. If it's not correct, please correct me when you are speaking, Dr. Sophie. So without further ado, I will stop sharing my screen and give the screen to Dr. Aniko Karozhi.
Okay, thank you very much. I'm just gonna change this still. Okay, so good afternoon, everybody. Thank you very much for the nice introduction and for inviting me to be part of this webinar. It's a real pleasure. And as Misha mentioned, I will be talking about my research line on early life stress-induced cognitive decline. And in particular, today, I will be telling you about the role of neuroinflammation and fatty acids in this. So just to begin with, I would like to highlight that the first 1,000 days of life are key determinants of mental health. And maybe this is something that most of you know, but for the first 1,000 days of life, we really mean from conception up to about the first 2 years of age. And this is a critical developmental period during which, indeed, if exposure to adversity occurs during either prenatal or postnatal period, this will have lasting consequences on the risk that one will have on developing a psychopathology later in life. And what is also remarkable is that when we look in the epidemiological literature, we can see that these early life adversities can take many different forms. So it can be, for example, in the form of maltreatment or in the form of malnutrition, etc. And what is remarkable is that actually, even when these adversities take such different forms, the outcomes or the phenotypes observed in these individuals are actually remarkably similar. What is also important to mention is that these psychopathologies are often comorbid with metabolic disorders, and also that these early life adversities not only lead to risk to develop these psychopathologies, but also these metabolic derangements. And the reason I'm mentioning all these different intersections is because it helps us, or we hope that it will help us understand a little bit better the mechanism underlying this long-term programming and how these early life stress-induced lasting effects take place. So indeed, in the lab, we try to understand what are then the key elements in the early life environment that determine these long-lasting phenotypes, while these key elements can be very different, while the phenotypes are remarkably similar. And what are then the main neurobiological substrates, and how do these different elements interact with one another? So some years ago already, we came up with this hypothesis, thinking that early life stress, which can then, as mentioned, can take many different forms, will impact on the organism as a whole. And this schematics is not meant to be complete, but here we represent the focus points of the lab. So focusing on stress regulation, on the nutritional and the metabolic profile and the inflammation, and trying to understand really the intersection and interaction between this system, and how then these impacts will ultimately lead to the alterations in behavior, so in the cognitive dysfunctions and the anxiety phenotypes that are being observed. And in particular, today, I will talk to you a little bit about the underlying neurobiological mechanism, and we focus on different aspects, of course, of the neurobiology, on astrocytes, on the synapses, and on microglia. And today, in particular, I will talk to you about the microglia part. And then, quite straightforward, really, is what role nutrients play in this, and can we use the knowledge that we have about nutrients on nutritional interventions that we could apply in this context, and whether we can protect against the lasting effects of early life adversity and protect against these cognitive dysfunctions and anxiety later in life. So the mouse model that we use in the lab is a very well-established mouse model of chronic early life adversity during the postnatal phase. So mice are exposed during the first postnatal week to an impoverished environment. What you can see here in this schematic is that indeed these mice are placed on a mesh and they receive no bedding material and just a reduced amount of nesting material. And this is distressing the mothers. It leads to a fragmentation in her care, and therefore also stress in the pups. And what is remarkable about this model is that the pups and the moms are together, making it a little bit more ecologically similar also to the human conditions, where often the mother is present but just unable to provide the care that she would want to provide. And what we have seen by us, but also by many, many other groups, is that this indeed leads to changes in brain plasticity at the level of neurogenesis, at the level of microglia, of astrocytes. And I will show you a few examples of this, and changes in behavior on the long term, making this a really nice and translationally valuable model to understand the mechanism of how this programming takes place, and also to use it for testing possible interventions or proof of concept interventions. So I will focus today my talk on glia. We have focused in the past also on astrocytes, and you can see in these papers, and if you're interested in it, you can go ahead and read about them. But as mentioned, today I will focus on some of our most recent work on microglia in particular. Microglia are the macrophages in the brain. I think that most of you are familiar with this. And what we have shown some years ago is that indeed this early life adversity leads to lasting alterations in microglia, both in normal wild-type mice, where we, for example, just a small example I show you here is that there is an increased phagocytic capacity, so an increased CD68 expression in the brains of mice that have been exposed to early life stress 4 months before. So at basal level, these mice show somewhat more inflamed brain or more reactive brain. And what we see here on the right side of this panel is that this is mice that are either transgenic for APP, so Alzheimer's disease, with the chronic accumulation of amyloid that are controlled or exposed to early life stress. So this context really indicates that there is of course, a chronic challenge in the brain because there is accumulation of amyloid chronically. And what we see is that, as expected, indeed, the APP mice under a control situation already exhibit an increase in microglia CD68, which is to be expected, of course. But that in addition to that, the early life stress exposed APP mice have a super exaggerated response to this, indicating that maybe indeed the microglia from mice that have been exposed to early life adversity might be hypersensitive or in any case differently primed to respond to challenges. So this is the question that we decided to follow up in the study that I will show you today. And to simplify our life, of course, this extensive long-term studies with APP mice and the crossbreeding, that's very consuming. So we decided to simplify a little bit the design, use only wild-type mice, and expose them to an acute LPS challenge to really understand how does early life stress affect microglia and how does this exposure affect the capacity of microglia to respond to LPS challenge. And what we did, it was very straightforward. We had mice exposed to this early life adversity or control, of course. And then at postnatal day 9 and at postnatal day 200, we dissected the hippocampi of these mice and sorted out microglia with a FACS sorter, and then looked at the with RNA sequencing to the gene expression profile. This is work that we did in collaboration with Groningen, with Professor Bart Egge. And what we found is actually really intriguing and interesting. So what you can see here is a 4-way plot. And here you can see the comparison basically of control animals P200 to P9 and the gene expression comparison of early life stress animals P200 to P9. So basically trying to tease apart what is the aging related trajectory or changes in gene expression from P9 to P100, 200, and how is this impacted by early life adversity. And what we can see in gray, you can see it in the Venn diagram or in the 4-way plot, is that indeed there is, of course, a large amount of genes that are shared between control and early life stress animals, as you would expect. But there is also a considerable amount of genes that are differentially expressed between these two ages that are different or exclusive depending on your early life stress exposure or not, kind of indicating that these microglia take a somewhat different aging trajectory, if you will, because of this exposure. So of course, then we were interested, how is this change affect the response to LPS? And what you can see here is a similar way of plotting the data. Again, in gray, you can see the shared genes, and in orange and dark red, you can see the ones exclusive to control animals or exclusive to early life stress animals. And what you can see is that next to the shared genes in response to LPS, which are actually the ones that one would expect, we can see that there is a large number of genes that are differently impacted by LPS depending on your early life adversity. So it's really interesting. Basically, it means that early life stress exposed animals, when then stimulated by a second challenge with LPS, they will exhibit a different gene expression profile within the microglia, also suggesting possibly a very different response. In particular, for example, what comes back quite frequently now in our analysis is that in the early life stress animals, this extracellular matrix seems to be more involved than cytoskeleton. While, for example, in the control animals, axonogenesis and synaptic transmission related genes seem to be coming better, more forward. So our next question that we wanted to understand was, okay, are nutrients in any possible way involved in this process, in this early life adversity induced programming? And of course, why nutrients? Well, I think in particular, of course, they are building blocks. We know that they build up all the membranes and they are really important energy source. But what is also intriguing about nutrients, of course, that they are key signaling molecules. And this is the aspect that I like to touch upon a little bit more in detail today. And while my talk is going to be really primarily preclinical, what maybe was already also clear also from the introduction of Sophie, for example, is that there is a really big boom in interest in the nutritional psychiatry field. So how we can modulate mental health with nutritional strategies. And this is of course extremely attractive, but as you go in the literature and you see the studies that are available, we also realize that we still need a lot of evidence, because currently we just lack mechanistic understanding and causality in these studies. So I think this field will profit a lot from further understanding of specific mechanisms. And I hope that my work actually contributes to these bits a little bit. So the questions that I will be addressing is whether early life stress alters the nutrient levels in the brain, whether nutritional intervention can prevent these early life stress-induced effects, and then if so, then what could be the potential mechanism for this? So the work that I will be presenting has been done mostly by 3 talented PhD students, Kitty Yam, who already moved on already a few years ago, Kitty Reims, who's going to, she's now in the phase of finishing her thesis, and Marilyn de Ebbing, who finished about a year ago. And the nutrients that we focus within the group are mostly the essential fatty acids and the essential micronutrients. And with the word essential, it's key to highlight because that means that this needs to be taken in from the diet. So it's not something that we can synthesize within our own body. And but today, of course, I will talk to you about, in particular, the work on the fatty acids. So the fatty acids, I think you are all familiar about the importance of omega-3 and omega-6. We hear a lot about it, and that we have the urge of needing a lot of fish and nuts and olive oil, and that it's much better than corn oil, for example. And what is important to realize is that, of course, these fatty acids have really important properties for brain development. They are key for membrane properties. For example, the membrane fluidity and how well certain receptors get expressed. For the process of neurogenesis, both during development, but also in adulthood. They are really important inflammatory modulators, and I will tell you a little bit about that in a moment. And what I would like to highlight is also that is actually the ratio of this omega-6 and omega-3 fatty acid that is really important. So for example, if you are having a really unhealthy diet with really high omega-6 content, then maybe it's not sufficient to just take supplements of omega-3 to compensate for this, because they will ultimately compete for the same enzymatic processes, and thereby you will still not manage to get back your balance. So it's really important to keep this ratio of omega-6, omega-3 within a certain value. In particular, I think also to make the link with microglia, this is a really nice schematic from a previous review by actually the group of Sophie. And what you can see here is that, of course, the polyunsaturated fatty acids and their derivatives, which they also refer to as SPMs or oxylipins, as Sophie will tell you a little bit more in detail. And this is just a nice schematic showing how these impact on several processes that are relevant for microglia functioning, such as like the inflammation, the phagocytosis, and the motility of microglia in particular. And this is just to show a really specific target mechanism of how certain dietary supplements with this PUFAs could be acting. So some years ago, we published a work where we actually wanted to test exactly this. So how would an early dietary intervention, where we give a diet that increases the availability of this omega-3 fatty acid, but by actually playing around with the ratio of this omega-6, omega-3 during the early life stress exposure and a little bit further to cover all the full developmental period, how will this act? Will it protect, or would it be able to modulate the lasting effects of early life adversity on cognitive functions and on the measures of brain plasticity that we observed? So very first, actually, we questioned whether early life adversity alters hippocampal fatty acid composition. And that was indeed the case. We see here that there are changes at all different omega-6 and omega-3 fatty acid levels. For example, here you can see that there is a reduction of DHA. This is actually quite remarkable because the brain is extremely well protected for its fatty acid content. So to be able to see any differences at all, even though they might seem small, that actually can have a really big biological impact. So next, when we gave this diet, what happened? When we gave this diet at postnatal day 9, so before the end of this dietary intervention, we sacrificed the animals to test their fatty acid content. And what we see is that indeed, we managed to restore, for example, here you can see for the DHA example, we managed to restore this DHA deficit that was observed earlier with an increase in the brain. So this is again quite remarkable because the diet is fed to the mothers who via their breast milk then feed it to the offspring, and thereby, and this is what we measure then in the brain of the offspring. So this is a really interesting finding. So what about the long-term effects? What we see is that indeed we are able to restore or to prevent the cognitive deficits induced by early life adversity. So what we see here is, for example, a deficit by early life stress in the black bar in the object location task. And what we see is that the animals that have been receiving the supplemented diet early in life, several months later, do no longer have the same deficit. Here, this is just an example on the object location, but in the paper you can see we find a similar beneficial effect also on object recognition and also on the Morris-Water maze task. And what about the brain plasticity aspects? So when we looked at neurogenesis, for example, so this was the survival of BRDU-injected neurons, a marker for neuronal cell survival. And what we see is that indeed we see a reduction by early life stress, as we would expect and as we have shown also earlier, and that this reduction is completely restored by the diet that was given early in life. And similarly, when we look at the CD68, just as I showed you earlier, also in this cohort, we find this increased CD68 by early life adversity. And we see that this increased CD68 expression is no longer there under the supplemented diet. So with this early diet, several months later, when the diet is no longer in circulation, we are able to restore cognitive functions and prevent the changes in brain plasticity that are occurring. So this, of course, made us really excited and made us even further more curious on what exactly does this diet do and how does it exactly work. And to dive further into this concept, what we did is to set up a new study with a really similar design. But in addition to this, we included an LPS challenge at the end, or saline, of course, to try to understand what happens more at the molecular and biochemical level in the brain in response to this diet. So what we did, of course, it ends up being a quite complicated study design with 8 groups, because you have the controls and the early life stress with the 2 different diets, and then with saline or LPS. So it ends up being an 8-group design, which is quite complex. And we looked at the peripheral cytokines, we looked at the gene expression profile in the hippocampus, and at the broad lipidomic analysis in the hypothalamus to have a view on what would this diet do. So first, looking at the cytokines, what we see is that, so to mention, the LPS dose that we gave was a relatively high dose of 5 milligram per kilogram, and the animals were sacrificed 24 hours later. In hindsight, we probably would have chosen a milder dose or a smaller dose, but at that time, we really cared of having the security of a dose that would lead to an inflammatory changes within the brain, which we definitely saw. But as you can see here with the cytokines, we have kind of an overshoot. Nonetheless, despite this high dose, we still see some specific effects, well, of course, of LPS overall, but also of early life stress, and also some interactions where indeed we see that early life stress exaggerates this response to LPS. And there is even some interactions with the diet, which doesn't seem to be, however, too prominent at this stage. So what about the gene expression? How does the diet impact on gene expression under basal condition and in response to LPS? And is this in any way impacted by this early life stress exposure? So what we did was a microarray. This was some years ago, so we didn't do the RNA sequencing at that time. But what you see here, this is simply a comparison of the control animals under the high or the low ratio fatty acid diet. So the low diet, just to remind you, is the one that was beneficial in rescuing the early life stress-induced effects. And what we see here is actually quite a nice confirmation maybe of our dietary intervention. We see several GO terms popping up, and in particular, I like to highlight the long-chain fatty acid transport and the long-chain fatty acid catabolic processes, kind of indicating that indeed these fatty acids have reached the brain and changes also gene expressions that are genes that are related to their processing. And what happens with this early life stress and in particular. So what we see here, it's quite interesting. What you see, these comparisons, of course, it's a bit complex, but here you have the high diet and the control early life stress comparisons. And here you have again control early life stress, but under the low diet, kind of trying to tease apart what is the impact of the diet on the early life stress response. And what you see, it's remarkable. So the gene expression profile in the hippocampus, it's very selective and very different in response to early life stress, depending on the early diet that was consumed. And when we look at the Go terms that are included in these genes that are impacted, we see that indeed they are also quite different. So we have here more related to biosynthetic process, mitochondria, and generation of neurons. And here under the low diet, we see more an extracellular matrix and cell migration. So keep in mind, so under the low diet, these early life stress animals no longer exhibit these deficits that the early life stress under the high diet exhibit. So these Go terms or these genes can kind of give us a leeway on the possible targets that could be taking a role in how this diet exhibits its beneficial effects. So what happens, of course, in response to LPS? That was the next question. And what you see here, so these comparisons indicate here is the saline versus LPS, and here is controls, the control animals responding to LPS under the high diet, and here is the early life stress animals responding to LPS under the high diet, and here below are the same comparisons, but under the low diet, so the beneficial diet. And without going into much detail, I thought what was remarkable is that, for example, also in response to LPS, just like at the basal level, the early life stress animals under the low diet again show changes in genes related to extracellular matrix. So this is definitely a target that we are currently following up. So what happened to the hypothalamic lipid profile? So we see these gene expressions that depend or that are different depending on your early diet, depending on your early life adversity, and there is of course a response to LPS. So we were wondering, can we find this back biochemically at the structural level in the lipid profiles in the brain? So very first, this is a comparison of just of the lipidomic analysis of just the effect of the diet, so in control animals. And what is nice to see is that, as expected maybe, but I don't know, this early diet several months later leads to changes in the lipid profile in the brain. And what we see is that in particular, for example, there is a reduction in lipid species that are implicated in inflammation, like the ceramide, for example, that are implicated in inflammation and stress. Of course, in addition to this, we were also wondering if early life stress would impact on this lipid profile and if the diet would do something to that. And you see that there are not that many lipids actually being affected, but that when they are affected, these are different in early life stress animals compared to the control animals. Again, indicating that the early life stress kind of sets the brain to a different trajectory, impacting on how the lipid profile will look like. And then when we looked even in response to LPS, which was even a group that we were doubting if to include or not, and we kind of included out of completeness in reality, but what we find was quite remarkable. So even 24 hours after LPS, we see that the lipid profile of the brain is different compared to the saline group. And we are still trying to understand what exactly can be going on within such a short time frame after LPS to lead to such big changes in this lipid profile. And then, of course, try to understand how the different groups, and we are still trying to tease this apart. We have all the different comparisons of the control animals in response to LPS and the early life stress and under the different dietary conditions. But what you can see here is that there is, of course, some overlap between the groups, but there is a lot of exclusive lipids that are affected depending on this early life condition, kind of maybe starting to give us a hint on how this could be happening. So I think in conclusion, I showed you that microglia gene expression profile is affected by early life stress on the long term, and it impacts on how this response, these cells respond to LPS. That an early dietary fatty acid diet protects against the effects of early life stress on cognition and on different forms of brain plasticity, for example, neurogenesis and markers for microglia. That central gene expression and also central lipid profile are affected by these early life conditions on the long term, and also how then the system responds to an acute immune challenge in the form of LPS. And what is then still now currently ongoing is that within the same samples where we measure the gene expression, we are now currently looking at the microRNA expression and integrate these to try to even further understand and dissect how these gene regulations might be occurring. And next to the lipid profile with this broad lipidomic approach, we are also looking at the specialized pro-resolving mediators in collaboration with HiSCOI and trying to integrate again the lipidomic and this pro-resolving mediator profile of the hypothalamic in response to these exposures. One last thing that I would like to mention is that, of course, we are intrigued of how the stress would even lead to a nutritional deficit. And one of the hypotheses that we have is that the breast milk composition of the mother might play an important role. And in order to study this, we have set up actually the Amsterdam Mother Milk Study together in collaboration with Professor van Houthoever. And we have now the first data that indicates that indeed stressed mothers exhibit different DHA profile in their milk. And we are now studying this further and also looking at other components in the milk. But this is quite promising, and it might really help to think of possible specific supplementation for mothers that might be in stressful conditions. And that in addition, next to the possibility of the milk option, it's also possible that the adult individuals that have been exposed to early life adversity might exhibit different food choices. And this is a preclinical study that we recently published, where we indeed suggest that early life adversity exposed females, when they are exposed to a stressor, they will end up consuming much more fat compared to their peers, also indicating some or giving some good hints in that direction. So I think my take-home messages for today are that, of course, animal models of early life stress are really important and translationally very valuable to get insights into the mechanism, that alterations in microglia are potential substrates for these ELS-induced vulnerabilities, that nutrients play a key role next to stress hormones and sensory stimuli and other aspects that have received a lot of attention in the past, that nutritional interventions could be a new potential avenue to protect against this early life induced vulnerability. Of course, we still have a lot of work to do also to tease apart possible other time windows and nutritional composition for this. That nutrients can contribute maybe in the search of biomarkers for those that might be vulnerable. And so we need just to understand much more about the mechanism and the causality to optimize the current nutritional strategies. And of course, I would like to thank all the group and that did the work. So the PhD students in the group, and in particular, as I mentioned, the work that I presented today is mostly work from Kitty Yam, Maralinde Abbing, and Kitty Dames. And of course, the collaborators from the different groups and the funding agencies. And of course, I'm happy to answer any questions if you might have any.
All right, Aniko, thank you. Great presentation. So we only have time for a couple of questions and some short answers, hopefully. And let's start with Bert at Hart. He asked the first question is there a role for stress hormones, so like the corticosteroids in the early stress phenotype induction?
Yeah, of course. Yeah, I mean, when I stepped into this field of early life programming, then by that time, the stress hormones have received a lot of attention. And so there are several studies showing, for example, that also by colleague Harm Krugers, but also by many others, that antagonists of glucocorticoid given during sensitive period of time can also be exhibiting some beneficial effects on the phenotype. There is, of course, increased corticosterone at this postnatal day 9, so there is definitely exposure to corticosterone. I think the hypothesis that we have in the lab is that there might be indeed an intersect between this nutritional message and the stress hormone message, and that this might, the signaling of these might somehow come together, and that you can maybe mitigate some of this glucocorticoid increase with the right nutritional strategies. Of course, glucocorticoid is really strictly related to energy and energy levels. So in that sense, the right nutritional input can actually influence and interfere with this.
Eric, would you like to?
Yeah, I see some interesting questions. I have a few myself as well. I think that the last one that came in is also of interest by Arnoldo Castro, about if you see perhaps a change also in the ratio of glia cells versus neuronal cells due to this early stress, and whether this may also affect how the glia cells, especially the microglia, how they may behave. And it is also a consequence of levels or of ratios of different cell types in the brain. Yeah,
so I think this is very interesting. So I think we didn't measure this systematically enough. I think within our sorting experiment, we did not seem to see that. So of course we dissected the hippocampus and we sorted the microglia, and we didn't seem to pick up on necessarily different amount of microglia being isolated, but this is quite a rough measure. So maybe this is something that we can look further into detail. So far within our stainings and our analysis, we did not come across this, but I have to say this is not something that we directly targeted. Yeah. Good point.
And I have one question myself, because the focus of the group is on cellular molecular mechanisms. And then I have it easy because I'm a cell biologist. I look at single cells. you look at the whole brain, but you even look at whole organisms. But the effects that you see, for example, with the cytokines or also the effect of early stress might just mainly happen at the intestine, the uptake of fatty acids from the food, and that it is mainly happening there. And what you see in the brain is only a consequence. So I'm very, very blunt here, but.
No, I think that's a very good point. So like, I think most of the time when we think of like, okay, how are we getting to a different fatty acid or nutrient composition in the brain? We have seen also examples of other nutrients being altered in the brain. And I think I always think of it in 3 blocks, like either it comes from the mother, like for example, from her milk, or indeed of how it's being absorbed, or that maybe a stressed organism has simply different need that remains unmet. And probably it's rather the combination of all 3 of these things. And I try to address each block. So we looked at the milk and we see that there are some changes. For the gut microbiome, we did a study in particular on a similar cohort without the LPS, but with the same dietary intervention. And we looked at the gut microbiome. And we see, of course, some changes at the gut microbiome level. And we also intend to look at, of course, at the maturation and the inflammation within the guts. Yeah. So this is also happening. And yeah, the need part is a little bit more difficult, but I think that's a little bit addressed by the fact that when you do supplement it, you see that you can reverse the effect. So it's possible that that's also playing a role. Yeah.
And you're making crosses with APP mice and also LPS. So what would be the next challenge? You see the question of autoboidins. Would you, for example, combine it with a mouse model with the LDL receptor knockout or the APO knocked out? What would be your favorite next step to look at mechanism?
Yeah, I think there are many different options, and I think the challenge depends on the question. So I think what we realize more and more is that to understand the effects of this early life adversity, you need indeed to challenge the system in certain ways to unmask latent effects, so possible latent effects. So we also do in the lab, for example, when we are more interested in metabolic aspects or other aspects, we do additional Western style diet challenges, and we can think of crossing also with other indeed genetic lines. Yeah. But also within the APP and so like that whole research line on the neurodegeneration, we see some really interesting age-dependent impacts of this early life adversity on how the progression of Alzheimer is. So we are really intrigued in trying to understand exactly what is there the basis of this age dependence. So like at 4 months and at 10 months, there are really different changes in the brain, and we don't understand the substrate of that yet. So I think we're first trying to tease that apart, and also to see if this diet can be of benefit also in that context, because now of course we separated the two, and because it's so complex, but I think I ultimately aim at converging these.
Would you have an off-record advice for the daycare centers and young mothers? There's a question by Arjan Brustad that you have these different ratios of omega-6, omega-3, and you have all kinds of different ratios. What would you have an advice here?
I mean, not specific yet, because of the fact that these studies, of course, are preclinical and they are based on the requirements of these rodents. But I hope to get a bit of a better insight of this from this breast milk study that we are now doing. And I hope that will give a little bit of a better insight on what are the expected doses. So this is a relatively small study, this Amsterdam mother milk study, but we also did it on a much larger scale under a different umbrella with more than 2,000 mothers. And I hope to get an even better insight on how generalizable is this effect on nutrients and how related it is to lactation stage, because of course this study is really on early lactation stage. But this will be the ultimate goal, to be able to give some more evidence-based advice, because now there is no such a thing. So yes.
Another word of advice for the people that love fish. I see Jinte, she wants to go to the herring shop immediately, but are there also negative effects of all the fatty acids?
Yeah, I think, well, there is of course like the whole sustainability issue of the fish consumption. So I think it's definitely important to think of other sources, of course, of these beneficial fatty acids. And I think what is also important is this ratio, so this balance. So I think it has to come. It's not enough to just eat herring on top of like a really unhealthy diet, but probably we need to interfere on all other aspects as well. But this is what I meant with this last line on the biomarkers, that I think right now everybody might have a different baseline, and I think maybe we need to first understand what our baseline is before we can go on and think of how to best supplement and how to move further. And for sure it's more complex than this. It's not only the omega-3s and the omega-6s either, so it needs to be a more integrated approach, I think, to do something with this. Yeah.
All right.
Thanks so much for the nice presentation.
Yeah, thank you for the presentation. We need to move on.
Thank you.
So, Sophie, please unmute and share your screen and start your presentation.
Thank you. Well, thanks a lot for the invitation. It's a great pleasure to be here. And so I'm happy to present some part of the work that we are conducting in the lab. So you heard a lot before from Aniko that the diet is really important in the development of mood and cognitive disorders. And what is well described now in humans is that obesity, diabetes, but also the consumption of Western pattern diet is really a high risk factor for mood and cognitive disorders. While on the other hand, the consumption of diets that are called prudent, such as the Mediterranean diet or the Nordic diet, are more protective and with less risk of developing those disorders. And we get interested in some nutrients specific from those diets. And you heard a lot about omega-3. So I'm sorry, I'm going to say things that Aniko said before, but we get highly interested in omega-3 a long time ago. And as you can see on this map, and Aniko mentioned it, so the level of omega-3 in the world is very different in terms of intake, but also in terms of accumulation in the body. And here, this is a map showing the level of two of the main omega-3 found in the body that are EPA and DHA. And according to the level of EPA and DHA in red blood cells, so the amount is considered either as adequate in green, while it's very low, and non-adequate in red. And you see that many countries do not reach the right level. So what are those omega that are called also polyunsaturated fatty acids? Well, there are two main families. Again, Aniko mentioned the omega-6 family and the omega-3 family. And what I would like to pinpoint here is that the dietary source of those two forms is really very different. And the form coming from plants that are the linoleic acid and the alpha-linolenic acid for omega-3 are really coming from distinct sources. And actually, we eat a lot of oils coming from sunflower, corn, and peanut, and less oil coming from walnuts, flaxseed, and rapeseed, meaning that the ratio in omega-6 and omega-3 that was mentioned by Aniko is really not balanced, because we are supposed to eat 5 of this kind of fatty acid coming from omega-6 for 1 omega-3 coming from those oils, and actually we're more close to 15 to 1, and in some country 100 to 1. So those fatty acids are essential, meaning that the body cannot produce them. So we really need to have them from the food and from these plants. Once we eat them, they are metabolized into what we call long arachidonic acid in the main long chain form from omega-6, and EPA and DHA are the main long chain form coming from the precursor omega-3, sorry. But what is quite interesting is that we can have them from the food. So this EPA, DHA is found in fatty fishes and some microalgae, and we mentioned before that this is the kind of new sources, dietary sources for the omega-3, and arachidonic acid is found almost everywhere in all the Western diet kind of food. So again, as mentioned before, those forms of omega-6 and omega-3 are really highly incorporated into the brain, especially during development. And DHA is the main omega-3 that is found in the brain, and arachidonic acid is the main omega-6 found in the brain. And those forms incorporate in the brain, again, coming from the maternal food before birth and after birth, before reaching a kind of plateau. And concerning the food, it's really important that what the mother will have in terms of omega-3 and omega-6 to get this incorporation in the brain of the baby, of the fetus, and after weaning, after birth from lactation. But then after, we have to keep a high level of consumption of this omega-3 and omega-6 to maintain the plateau, and especially during aging, this is very important because there is a kind of decrease in the aggregation of DHA, especially with aging, and so that could be maybe compensated by the food. So there are again in the literature a strong link between mood and cognitive disorders and the polyunsaturated fatty acid metabolisms and the amount that is found in the diet. And what has been really clearly described is that there is an inverse relationship between fish consumption that is rich in DHA and EPA and the prevalence of depression and cognitive decline. Also, some omega-3 level in the blood and in the brain of depressed subject that has been described as really lower as compared to healthy subject. And this is the same also in the brain of aged subject with cognitive impairment. And of course, there are many, many animal models showing that the decreased dietary intake of omega-3 alters the level of omega-3 in the brain and increase emotional behavior impairment and memory impairment. What is quite interesting is that there are also many trials trying to give back this omega-3 to subject or to patients suffering of depression. And actually the results were really not really good, except in very specific type of subjects. In particular, the patients, the depressed patient exhibiting inflammation. So in this case, the supplementation with high level of long-chain omega-3, DHA or EPA are more potent. So there is an effect, especially with EPA that is now thought to be really the active form for depression, and in combination also with antidepressants. So it seems that omega-3 can be used as an add-on strategy to improve the effect of antidepressants. And also concerning Alzheimer's disease, the results are quite poor. It seems that the level of DHA that is given as a supplement can improve a bit some, a bit Alzheimer's disease, especially in patients not bearing the amyloid, amyloid load. But concerning cognitive decline in aged subject, multimodal intervention with omega-3 may improve cognitive decline. So concerning the mechanisms, and again, Aniko said how it is important to understand how those fatty acids can work. What is known is that they incorporate into a membrane, so they give a lot of properties to this membrane, especially fluidity, and it influences the functioning of several receptors and signaling pathway, also some channels. But they can also be released out of the membrane thanks to specific phospholipase. And these three forms of fatty acids, so arachidonic acid for the omega-6 family, DHA for the omega-3, will give rise to new metabolites. Some are called oxylipins, and they influence lipid signaling and they control inflammation. But they are also precursors of other molecules such as endocannabinoids that are high potently regulator of synaptic plasticity, and they can act directly on several receptors to regulate gene expression. And we get particularly interested in oxylipins, those derivatives of PUFA, of omega, that control inflammation. And to make a very complicated story quite short, arachidonic acid is a precursor of many compounds that are mostly pro-inflammatory. So they derive from the cyclooxygenase pathway or the lipooxygenase pathway, and all these compounds are mostly pro-inflammatory. But there are some compounds deriving from the cytochrome P450 pathway that are anti-inflammatory. So they are coming from arachidonic acid, but they are anti-inflammatory. The thing is that there is, when they are degraded by another enzyme, the enolase, then they get pro-inflammatory again. So there is a kind of balance here involving several enzymes leading to inflammatory oxylipins from arachidonic acid. On the other side, the DHA, but also the EPA derivatives are more pro-resolutive and anti-inflammatory. They are mainly derived from the lipooxygenase pathway. And we are talking here about resolvin, neuroprotectin, maresin, and there are more and more of these compounds that are discovered. And most of them are anti-inflammatory and pro-resolutive. So again, the balance between arachidonic acid and DHA and EPA is really important in the synthesis and the proportion of this derivative, these oxylipines, that will be either pro-inflammatory or pro-resolutive. And so in the brain, this is less well known, and we heard a lot about microglia, and actually we get interested in understanding how the amount of omega-3 in the diet can influence neuroinflammatory pathways and targeting microglia, and whether it could regulate this activity. We're also interested in synaptic plasticity, but I won't talk about this today. And neuroinflammation is really important, as I said before, in mood and cognitive disorders. So we believe that the relationship between the amount of omega-3 in the diet and these neuroinflammatory pathways are important for mood and cognitive disorders. So actually, one thing that we did first is to describe the amount of omega-3 and omega-6 that are present in microglia cells, because as I said before, DHA is the main omega-3 found in the brain. EPA is really poorly detected. It's really high at the periphery, but poorly detected in the brain. But when we look at the level of cells, then the amount could be different. So this is what we did here. We submitted mice to different diets. So a diet that is deficient in omega-3, a diet that is balanced in omega-3 and omega-6, and a diet that is enriched in EPA and DHA, so the long form of omega-3. And we measured in microglia that were sorted out of the brain of these mice, the amount of omega-3 and omega-6 found in these cells. And actually what we found, and it was really a big surprise, is that those cells have a lot of EPA as compared to DHA. And actually this amount of EPA poorly changed according to diet, at least this is not significant, while DHA amount increased with the amount of omega-3 in the diet. And the highest level found, of course, is associated to the consumption of a diet rich in DHA and EPA. On the opposite, arachidonic acid that is present in the membrane of these cells is poorly changed, but DPA, that compensates the changes in DHA, so this is also an omega-6, is present in the membrane at very low level, but is changed according to the amount of omega-3, so decrease when omega-3 increase in the diet. So really, this was really surprising for us to see that EPA was so important. And actually, so we try to understand whether DHA that was administered at the periphery could influence of pro-inflammatory factors that are known produced by microglia in the brain. So we use a very simple paradigm. We administrate LPS, that is an endotoxin, a bacterial endotoxin to mice, and we measure the production of several pro-inflammatory cytokines, IL-6, IL-1 beta, and TNF, that is increased after the administration of LPS. But when we co-administrate DHA in the tail vein, we saw a decrease in the production of some of the cytokines, not all of them, but mainly IL-6. So it seems that the DHA that is found in the blood can reach the brain and then act very quickly, because this is only 6 hours after the administration of LPS and DHA, really quickly decrease the production of cytokine in the brain. And another thing that we wanted to understand was whether when we change the amount of omega-3 in the diet of animal, we could change the amount of oxylipines found in the brain. So here again, we fed animals with a diet that is deficient in omega-3, and the other group was a group with, of animals fed with a diet rich in EPA and DHA, so rich in long-chain omega-3, sorry. And we treated again the animal with LPS and measured the production of oxylipine in the brain. And what is quite interesting is that really the amount of oxylipines found in the brain is highly influenced by the diet, but poorly by the inflammation itself. And this was surprising again, because all the LOX and the COX pathway is usually highly activated by inflammation. But the main factor influencing the amount and the type of oxylipin found in the brain was really the diet. And what we found is that when animals are fed with this DHA and EPA, there is a strong increase in the pro-resolving mediators coming from either DHA or EPA or from arachidonic acid. So the pathway, the CYP450 pathway that leads to these anti-inflammatory factors, pro-resolving factors. And in vitro, so this is a culture of microglia, when we treat the cells with LPS, we found that there is a strong increase in the expression of some of the receptors of those compounds, those oxylipins that we found to be increased in the brain of mice fed with DHA and EPA. So actually, the receptors for those compounds were highly expressed after a treatment, with LPS, so inflammation, and the LOX and COX pathway that are involved in the production of those anti-inflammatory resolving coming from DHA and EPA were also increased, suggesting that microglia could be targeted directly and produce their own oxylipins. And when we treat the cells, the microglia cells with LPS, there is an increase in the production of pro-inflammatory cytokine, here IL-6. And when we give the resolving D1, that is an anti-inflammatory oxylipin, this production is a bit decreased. And this effect, this anti-inflammatory effect of resolving D1 is blocked by the administration of a specific inhibitor of the receptor of this oxylipin on microglia. So again, suggesting that microglia are really able to produce their own oxylipins and that there is a kind of loop of action to control the production of pro-inflammatory factors directly at this level. So to sum up this first part, so what we found is that low dietary omega-3 level in the diet is really important for the regulation of oxylipins and and really the factors that are produced, these oxylipins that are produced in the brain are different according to the level of omega-3 in the diet, that those omega-3 target directly microglia and that really control neuroinflammation and that it could be through the oxylipins. So another thing that we were interested in, and again, Aniko talked a lot about that, is how can those polyunsaturated fatty acid control microglia during development. And as Aniko said, microglia are really important in the brain of the babies because it plays a key role in the way neuronal networks are shaped. So what is thought is that microglia are able to prune specifically spines that are not necessary during development, and they are using the complement system in order to recognize the synapses to remove and they phagocyte them. And what is known also is that in case of inflammatory event occurring either during gestation or in early life of the baby, there is an impairment in the microglia activity, and those microglia lose the capacity to selectively remove synapses, and then it's a bit messy, and then the neuronal network are not well developed. So we were wondering whether the amount of omega-3 and omega-6 in the diet could influence microglia activity in the same way that an inflammation can do it, and whether it could influence synaptic pruning and have an impact on several behavior, and in particular, some that are involved in neurodevelopmental disorders such as autism, for example, as microglia functioning is really impaired in these diseases, and that the link with omega-3 has been described in humans. So what we did is that we submitted again animals to a diet that is sufficient in omega-3, so the balance in omega-3, omega-6, as compared to a diet that is deficient in omega-3. So the diet was given from the first day of gestation to animals, and we studied the offspring at weaning. And what we found is that there is a decrease in the number of spines of neurons in the hippocampus of these mice that have been submitted to the diet that is deficient, but also a change in the dendritic arborization as seen here. And that there is also an impairment in the production of some of the factors of the protein that are synaptic proteins, that are again produced in the hippocampus. And when we measured memory, spatial memory, using a Y-maze test at weaning in animals, what we found is that there is some memory impairment, because animals that have been submitted to the deficient diet are not able to recognize the new arm that they are supposed to recognize, meaning that they have an impairment in spatial memory. So in collaboration with Marie-Ève Tremblay, we measured the amount of spines inclusion in microglia using electron microscopy. And what we found is that the amount of spine inclusion is higher in microglia of mice that have been submitted to the omega-3 deficient diet, suggesting that those microglia are more phagocytic, they phagocyte more spines. And actually looking at the co-immunolocalization of PSD95 and microglia, we also found that more of this spine protein in microglia, suggesting again that those microglia are more phagocytic. So we decided to study a bit more this phagocytic activity of microglia toward synaptosome. So what we did here is that we took out microglia of the brain of these mice, either coming from the sufficient diet or the deficient diet, and we submitted them to synaptosomes coming either from the brain of mice that had the sufficient amount of omega-3 or the deficient amount of omega-3. The purpose here was really to try to understand whether the deficiency in microglia was sufficient to increase the phagocytic activity, or if it was the crosstalk between deficient microglia and deficient synaptosomes that trigger really this phagocytic activity. And what we found is that the fact that microglia are deficient in omega-3 is sufficient to activate the hyperphagocytic activity of microglia, whatever type of synaptosome they are submitted to. So really meaning that the changes in omega-3 at the level of microglia is sufficient to trigger this hyperphagocytic activity. So then we studied the microglia. So first we described using this approach in collaboration with Oleg Butovsky that microglia have changes in their homeostatic signature when they are submitted to a deficient diet. So they have really a strong change in the expression of homeostatic markers, but also lipid markers and immune markers. So they are polarized into a different phenotype that seems to be more pro-inflammatory and less homeostatic. And then we studied whether those microglia have also a different profile in oxylipins. So again, we took out the microglia of the brain, and in collaboration with Richard Bazinet, we measured several of oxylipins. And what we found is that microglia coming from the deficient omega-3 diet have a particular profile in oxylipins, and they have more trioxylin, more HTE, and less so coming from arachidonic acid, and they have less oxylipins coming from omega-3, DHA or EPA. So we decided to study whether those oxylipins could be involved in the hyperphagocytic activity of microglia. So first we did some in vitro experiments where we exposed microglia in culture to several of those compounds, especially the HTE and so coming from arachidonic acid, so pro-inflammatory, and the other forms that are coming from the omega-3, DHA, and EPA. And what we found is that there is no clear effect of those compounds coming from omega-3 on the phagocytic activity of microglia, while 12-HTE seems to increase the phagocytic activity of microglia. So here what we measure is really the phagocytic activity of microglia towards synaptosomes. So we were wondering whether 12-HTT could be important, produced by microglia, could be important in activating this hyperphagocytic activity, and maybe playing a role in memory impairment and the synaptic network impairment. And so we administrate in vivo to animals bicalene, that is a flavonol inhibitor of the enzymes involved in the synthesis of 12-HTT. And actually what we found is that the phagocytic activity of microglia treated with bicalene is decreased, both in animals deficient and sufficient. And the hyperphagocytic activity of microglia described in the deficient omega-3 microglia is decreased by this bicalene, suggesting that 12-HTT could play a role here. And we could restore also the behavior of those animals. So when we administrated bicalene to perhaps before weaning, we could restore the spatial memory measured again with the Y-maze test. So here in animals that are deficient, the time spent in the two arm, the arm that they visited before and the new arm is the same, while as you see here, after the administration of bicalene, there is an increase of time spent in the new arm, suggesting that animals recognize it and so have an improvement in spatial memory. So to end, it seems that at least we found in this animal model that the level of omega-3 is really important to polarize the microglia during development, and that this high level of arachidonic acid in the brain and 12-HTT polarize microglia toward the hyperphagocytic activity that will play a role in the impairment of the formation of synaptic network and memory impairment. And I would like to end here and thank all the talented doc and postdoc that contribute to this work, especially Charlotte Mador, who is now a researcher in the lab, and Jean-Christophe Delpech and Quentin Lérol and Maud Martina, Lydie Morel, and Moira Rossito, and the collaborators, of course, and the funding. And I would like to thank you for your attention.
Thank you, Sophie, for a really nice presentation. Are there any questions from the audience? You can still put them in the Q&A, and maybe in time, Eric, do you have a question?
I have a question, but also see now a long question coming in from Arnoldo Castro.
Yeah.
So it's about the role of the effect of the PUFA diets on the mitochondria, because it might also be integrated in the mitochondrial membranes and thereby affect mitochondrial activity. Did you look into mitochondrial activity?
Unfortunately not, but this is a very good point. Yes, I mean, PUFA can influence microglia function, the activity of cells, and we know now that the metabolisms, of course, of microglia is very important. So those PUFA could act directly on this, but we did not have a look yet to this. It's a bit complicated, actually, to study mitochondrial activity in microglia, because those cells are really highly versatile. So it's a bit tricky. But yeah, that's a very good point.
And I guess also a bit similar question to also what Aniko got. Is it a direct or indirect effect? But then in the case with the PUFAs, they might also affect the white blood cells of the peripheral immune system. There might be crosstalk there. So then that they might also chat with each other and affect each other. Did you look into this, or infiltration?
Yeah.
Or?
Yeah, so we have no, in the last study that I presented, we have no infiltration of immune cells. So what we are looking at really is microglia, so the effect of PUFA on microglia. But of course, I mean, the peripheral cells are highly affected by the diet, of course. Those cells renew quite a lot, while microglia do not, or they poorly renew. I mean, they really, yeah, because I didn't say that, but they are coming from the yolk sac really early in life, meaning that they do not exactly have the same origin than myeloid cells, and they colonize the brain and they stay forever. let's say. That's why we were interested in the long-term effect of diet on those cells. And at the periphery, the myeloid cells, of course, are influenced by the diet. So the amount of omega-3 could be changed quite easy. In 2 weeks, it's sufficient to change the amount of omega-3 in those myeloid cells, but they are renewed. So if we change the diet, for example, so if we use a diet that is poor in omega-3 for 3 weeks, we will change the amount of omega-3 at the periphery, poorly in the brain. It takes longer. If we switch back to a good diet, high in omega-3, we'll change again the level in myeloid cells, but not in the brain. It's not sufficient. We need to give it a longer time. So really, even if, yeah, both cell types are impacted, they are impacted in a different way, and in a very short time at the periphery, very long time in the brain. So even if there are changes at the periphery, it cannot explain what we see in the brain because the changes are not the same.
The question from the cellular and molecular mechanism group, you do most of your work in mouse models, also even up to EM studies that you look at the pruning and the spines and the synapses. Do you have a favorite model that you can also mimic this with 2D or 3D with in vitro models, that you can look at the microglia and how they react on the PUFAs and also in interactions, branching, etc.? Is there a good model to mimic this in an in vitro setup?
This is very difficult. You mean how to reproduce this exactly in vitro?
At least so that you can study some of these mechanisms, because you feel more and more pressure that you have to go from mouse models also to more in vitro models. And now with the IPS models, you can mimic it nicely 2D and 3D. But I don't know if you have particular mechanisms in your lab or in your institute that you have.
So yeah, we are trying to co-cultivate microglia cells and neurons, but one important partner is also astrocytes, for sure. We need to really have them because it's crucial. So we know at the moment we mainly focus on this single cell, I mean only microglia, and what we plan now to develop is more iPSC and organoids in humans, because what we would like to switch to really is human. But the main problem in vitro anyway, whatever it is, I mean, 1 cell or 10 cells together, is the amount of omega-3, because all cell culture are so poor in omega-3. Actually, so if you look at the membrane of cells that are cultivated, the amount of omega-3 is so low. It's nothing to do with what is found in the brain. So the tricky part is really first to cultivate them, adding the right omega-6 and omega-3 to get the expected ratio between arachidonic acid and DHA or EPA in the cell membrane first, then to change the level by adding other kind of omega-3 or oxylipin or whatever to see the effect. So the real tricky part, in addition to what you say, I mean, to have a complex system in vitro, is also to have the right amount. Otherwise you do, I mean, no omega-3. So if you add some, there's a strong increase, a strong change in membrane, and the membrane start to have this curvative, because the level of insaturation is suddenly very high, but it's nothing to do with reality. It's just not good, to really study mechanisms. So that's a tricky part, actually.
I think because of time, but also I don't see new questions coming in. I would like to thank you on my behalf very much for your presentation. So thank you so much.
Thank you.
Yes, thank you. And of course, both speakers, really great presentations. Thank you very much. Thanks to all people that joined us. And you can actually watch the recordings of this session on our TN2 website. And please notice our next webinar on neurodegeneration on the 25th of November. Same time, same place. So the register is open, so please register, and see you all next time. Thanks for joining.